option even is. Yeah.
Is only.
Yes, if you need to pay to actually get.
It says recording unavailable. You're not allowed to record this video call.
But I don't think, because we are not the host. Yeah.
Okay.
We are not the host. Yeah. Okay. All right. Perfect. Okay. Thank you so much. Sorry about that. Mhm.
There we go. Mm-hmm.
That's interesting.
Way for him. Um, yeah, I think it's okay. No structure or whatever.
I guess you can't share.
Yeah.
You which compound you want to show?
Yeah,
Maybe I have.
I think I can share. Please, can you?
Oh, there it is.
Me. This is me. Oh,
You can see it.
okay.
That makes sense. Yeah, that makes sense. Okay.
Yeah. Mm-hmm.
So, Wen Yun, you should mute your phone if you don't speak.
Because this is my screen. Okay.
So I have a question about this. Maybe this is a bit of an aside, but wasn't Trovan a topoisomerase inhibitor? Is there any kind of like assay for topoisomerase? Ah, I see. Okay. Gotcha. Okay.
Okay, so you can go to the slide 3. No, I can yeah, structure the law. Well, the big difference between the Trovan and the 3 compound is the can you point out is the nitrogen and the phenol, if you can, and Eva, can you point out who? Okay, 文云，
依林现在是你吗？
you can
等一下，现在是好，
yeah, that's okay.
OK
Okay.
OK
Then you move to the Trovan, you can see the nitrogen and the phenyl group. Can you? Yeah.
呃，
Oh, okay, correct. Then you move to our compound.
our compound,
The
please.
same. Yeah. There's one more carbon is the CH2. The nitrogen and the phenyl, they have like some kind of rotation, right? They have some kind of the steric hindrance. But when we introduce the one more CH2, make it more flexible, okay, right? And then so that's why we guess they may did not have the antibacterial activity anymore because I I I believe the Trovan is like the topo 2 inhibitor, okay, to clear to kill the bacteria. So that's why we introduced the CH2 there, and then we found out it can maintain the Panx1 activity. So that I believe maybe our compound have the better selectivity than the Trovan. And then another the reason is we change the amino group on the left hand side. So you can move to the 5,3 the Trovan. You can move the Trovan. You can see that this functional group may be troublesome. So maybe cause the liver damage. But when we go back to the normal, the making drug like the mesopiperidine, so you can move to our compound, the same region, okay? So you can see that's why we replace this functional group instead of the 5,3 fused membrane. So that maybe can reduce the liver toxicity. Yeah. Yeah. Okay. Yep. Okay. Thank you.
Yeah, let's do it. Share screen. Can you see it? I think you recognize the slide. All right. Okay. So before we start, let me just say that majority of our work is not actually done on the human version of Panexin. For what we did so far, we used frog Panexin from Xenopus tropicalis, and it is because, at least in our hands, this protein is much better behaved, much more predictable, more stable. It's easier for us to purify, and it's easier for us to obtain in high yields, and we need those high yields to do things like cryo-EM, right? However, when it comes to things like drug binding, majority of the residues around the extracellular openings are very highly conserved among all the Panexins from different species, and they are basically the same between frog Panexin and human Panexin. So if we have a compound that binds around the tryptophan ring in human Panexin, I would expect it to also bind to frog Panexin. There might be some differences in affinity and so on, but it should interact with both proteins. So what I'm going to show you today, everything has been done on frog Panexin. So that's how we started. Yes, if you have any questions, just stop me. Yeah.
Well, so the first question was the answer is frog. So Adi used frog Panexin for his liposome experiments. And Susan, I think, can verify that. I'm pretty
Yeah,
sure.
that's correct.
And the when you say C-terminal deleted or C-terminal truncated form of frog Panexin, it depends where the truncation is. But if the truncation is the D357 or something like that, the very
350, yeah, 357 was the one, but Adi didn't use the deletion. He just used our caspase and cleaved naturally with caspase.
right. So the no endogenous activity. That's right.
Yeah.
No endogenous channel activity, no constitutive channel activity. My recollection is that can be activated by alpha 1. Yeah, I think you did that, Eva.
Yeah.
But Adi's was full length and then caspase cleaved.
Yeah, three. Correct. It doesn't seem to be accessible to the caspase, at least in our purification system. So when we cleave, we only seem to get the cleavage at the C-terminal one and not in the loop.
Yeah. All right. So first thing we try to do once we obtain your compounds is to obtain binding affinity in vitro, right? And the first technology we try to employ is called spectral shift. So basically, it's simple. You have your protein of interest. in our case, it's Panx1, and you label it with a fluorophore. And then you mixed it with fixed amount of your compounds in a dilution series. And what's happening is once your compound is binding, it causes I think I don't know if you guys hear that, but I hear a lot of
I hear a ton
like,
of,
yeah,
I'm hearing it
yeah.
also. Can we get, somebody needs to mute.
Yeah. All right. So basically binding events are recorded as a shift of spectra in either blue or red. And the KD is calculated by plotting the ratios of fluorescence intensities from the two wavelengths we're using, 670 to 650, and against the concentration of our binding partner. So it's quite simple. We quite like this technology. It uses very little protein, but unfortunately, as I said, you have to label your protein with fluorophore. So we tried to use it on all the compounds that you sent. However, we only succeeded with compound 35. With compound 62, we were unable to complete this assay because of low solubility. And with compound 70, there is actually some autofluorescence of this compound. So it just didn't happen. So the KD we obtained is slightly different. It's 27.6 from 3 replicates. But from what I read, like the difference in KD between those two different assays, like dye uptake in cells and in vitro assays, that's to be expected. It's a very different type of measurement if you think about it, right? The other problem here, as you look at it, this is from the 3 replicates, the error bars are huge. So it's not, I would take the value with a grain of salt, of course. But yeah, that was the first value we obtained. However, it was just for one compound. So we were thinking, what can we do for the others? And I found this paper where the authors measured binding of 3 different compounds. It's methylquinine, quinine, and quinidine to connexins, to a similar group of proteins. So they tried 2 different things. They tried tryptophan quenching because in their hands, the compound was binding to a spot in the funnel of connexin, which is like rich in aromatic residues. And as it's binding, it's actually quenching the native tryptophan fluorescence, which is exactly what is happening, we think, in our case, right? Because the extracellular binding site is very rich in tryptophans. So that's one thing to try. The second thing they did is they discovered that all those 3 compounds had actually stabilizing effect on the connexin 36. So basically, the more you add, the more thermostable the connexin 36 becomes. So how is it measured? So basically, we are measuring unfolding of our protein as a function of temperature. And Tm is a temperature where 50% of our protein is unfolded. So the more methylquinine you add, the higher the values become. So the more thermostable is the protein. So this is measured using technology called nano-DSF. And we actually also have equipment in the lab that is capable of measuring this value. So this was the next thing we tried. So basically, what we are doing is this time our protein is completely label-free. It's under native conditions. And we are basically detecting changes in tryptophan fluorescence as we are increasing the temperature. So here our thermal lamp, for example, was from 20 degrees to 110. So as you increase the temperature, you see that the ratio of 350 to 330 is changing. So in ideal world, if you have extremely stable protein, this line will be flat. And then at about 80 degrees, you would see a change. And this is where your protein unfolds. So frotpanexin is well behaved. However, you see it's slightly like creeping up as a function of temperature. But the majority of unfolding happens here around 80 degrees. So from the first derivative, you can calculate that the 50% of your protein is unfolded at about 78 degrees, which for a membrane protein is actually quite good. So what we try to do is we try to add increasing amounts of compound 35 and see if our melting temperature changes. And unfortunately, it does not seem to have this effect. However, the more you add, the lower your fluorescence gets. So basically, the compound 35, it's quenching your tryptophan fluorescence very significantly. At no compound 35, at 0 micromolar, we go to like 3,000. And at about 200 micromolar of the compound, we end at 600. So it's a very significant drop. So even though it doesn't seem, we can use the TM, the melting temperature. What we can actually do is use the fluorescence count at 20 degrees. So it's a pretty complicated technology, but here we use it as a very simple way. We didn't use this full measurement. We just took the values at 20 degrees, right? The fluorescence. All right. So if you plot it, for example, for compound 62, you see that at both 330 and 350 nanometers, we start with very high counts. We go to about 5,000. And as we increase the concentration of the compound, the values drop quite significantly till they plateau, right? So this is something we can use to calculate binding. So that's what exactly we did for all 3 compounds as well as for Trovan. So this is basically tryptophan quenching of our frog Panx1 in response to treatment with our different Trovan derivatives. The values we obtained are quite different from yours. They are much higher, but I think they sort of make sense. We had the highest affinity for the compound 62, about 8 micromolar, and from the initial values that you sent me, you provided 0.24. So it's a weaker binding, and Trovan was the weakest binder. However, there wasn't that much difference between Trovan, compound 35, and 70, unfortunately. However, like as I said, this is not human protein that you use. This is still frog protein. That's the one thing. And as you can see, at our beautiful binding curves, some points are like a little bit of an outlier still. So this assay can definitely be repeated with more points and more accurate, and hopefully would provide more accurate values. And finally, the best way of doing it is actually to use high-end fluorimeter, and this is what we are planning to do. We sort of use it in a strange way where we use nano DSF equipment to get fluorescence measurements, but I think it's a very good and promising start. But that definitely, like, there is definitely room for improvement to getting those values more accurate. Any questions? All right. Okay. Drag. Yeah. Yeah. So basically, yes. So basically, you have your ring of 7 tryptophans, and they have their own fluorescence emission, right? Which peaks at 350 and 370, right? And basically, as you add something which binds to the spot, that fluorescence changes. So basically, you change a local environment around the tryptophan rings, and they react. You cannot measure binding of anything to anything using this method because you often have drugs that bind to a site where there are no tryptophans, right? But we are in a situation which is, we are lucky, right? We have a lot of tryptophans in our binding pocket, and that they give us a very good signal. So that's what we are using here. Like, we are measuring basically local changes in the environment of this binding site. yeah. So basically, what? Please go ahead. Yes. So this is purified. We do all the quality control as we go through our purification. It's pure. It's homogeneous. There's like if there are contaminations, they're like very minor amount. And so far in our hands for different assays, the like frog Panexin behaves quite well. So like we do full characterization before getting to this point. Like we run gels, and so on and so on. So we are sure this is frog Panexin, but it's reacting to it. No. Yeah. I'm pretty sure it's 1 to 7. It's 1 to 7. Yeah. We'll get there. Yeah. Yes. Yes. So I will talk to it in I think next slide, but I'm pretty sure you have one molecule of drug binding to one heptamer. I think because when you look, it's yeah. I think it's probably one. If it is possible, maybe it's two. But basically, if you look at any published structure of Panexin, like the distance between two tryptophan side chains is like 10 9 angstroms. It's not that big. So, and that makes modeling more difficult. We'll get to that in a second. But yeah, I don't think it's 1 to 1. I think it's 1 to 7 or maybe 2 to 7 or something like this. All right. So we will use better equipment to actually perform this assay, preferably fluorimeter, but we also have in the lab, and hopefully that will make our error bars smaller and the values more reproducible. And like I said, it will be good once we have the assay set up to actually do it on human Panexin instead of frog Panexin. I think the values would be similar because that particular binding site is almost the same between the two. And what also would be good to have is to have a good negative control for the protein. So basically the tryptophan to alanine mutant of the 74, right? Because that should basically make the binding go away almost completely. So that will tell us whether what we see is a specific effect. All right, so that is all we have regarding in vitro binding so far, right? Okay. So now we are moving to cryo-EM. So just in a nutshell, what we are doing is we have our purified sample here in our institute. We put it on the grids, we freeze it, and we image it on our Titan Krios microscope. And here you see a bit of micrograph using different frog Panexin particles. And it's working very well for us in cryo-EM, especially the frog protein. It's very easy to process. And we are getting reliably to like 2.7, 2.8 angstroms. So far it's been working pretty well. So from that, you perform what we call image analysis. So you extract different frog particles, you perform alignment and averaging. From that, you get your 3D map, and then you can build your model. So that's the workflow. All right. So a little bit of background. So our frog Panexin is purified from SF9 cells, and we solubilize in DDN CHS. Then we exchange to detergent called GDN, which is a synthetic form of digitonin. And before we freeze, we do size exclusion chromatography to make sure that it's very pure and very homogeneous. And when you look at what was published so far for the frog Panexin, it doesn't really matter what the membrane mimetic is. It can be in nanodisk and can be in detergent, it always sort of looks the same, right? And that basal state we call inactive state, right? It looks about this. There is one exception to that rule, and it comes from Coatis' paper from 2024, and the PDB code is 9BZ8. So he published a map of procpanexin with what he called a C-terminal activating domain, which is different. So it's much wider in the cytoplasmic domain, about 10 angstroms. So the difference is pretty significant. And it has N-terminus inside the pore, because in our inactive state, we only see a few residues of the N-terminus, and it seems to be pointing like out of the pore. And so far, working on procpanexin, we discovered there are different ways of making it active. This activation can be caused by mutation mimicking phosphorylation or deacetylation, or also by adding some chemicals after purification, like for example, by oxidizing the protein. But what we like about procpanexin is, unless you do something drastic to it, it almost always looks like this. It's very predictable, right? You have to introduce some mutations or do something like chemically drastic to it to make it active. Right. However, for the sake of binding of those compounds, if they really bind around the tryptophan 74 and arginine 75, that particular region of the protein is the same between two forms, right? So extracellular domain is basically identical, only as you go towards the cytoplasm, the protein sort of swells. Does it make sense? All right, so that's for the introduction. All right, so other people have tried to get drug binding to Panexin before us, and it sort of doesn't work that well. So I think first attempt was in 2020 from this paper where authors claim they see CBX density around the extracellular domain, but it's a 4 angstrom map, and what you see is a blob. It's a blob. It's very hard to say what it is. And what is also very confusing with Panexins is, even if you purify it without any drug, even if you do not add any drug, very frequently there is some density here around those tryptophans, even if you have added nothing. And we believe this is like a very low unspecific binding site, and probably as the protein is expressed in the cells, different things from the cells sort of bind to it. And as you purify it and do your cryochemical analysis, you will see some noise around this place, right? So it's making your analysis more difficult. That was one attempt. The second very interesting attempt was with human Panexin, and that was adding probenecid, right? And in this case, the authors didn't see the actual density, but they saw an indirect effect of the drug, right? So human Panexin is different from procpanexin. If you purify it and don't add anything to it, don't do anything to it, you have wild type, most of the time it's already in what we call an active state. And Susan and I, our theory is there is some kind of post-translational modification happening it as it's expressed. It might get activated in the cells. It might be cell type dependent, who knows? But basically, authors got this active state of human Panexin, but as they added the drug, it went to inactive state, right? So you initially have your N-terminus inside, you add the drugs, and the N-terminus flips out, and it becomes what we call it, inactive, which is a very interesting effect, even though they didn't really see any significant drug density around the extracellular opening or anywhere else, really. All right, so those were the two attempts that I know of, like, and then let's move on to what we did. All right, so every single structure you see on PDB was processed with 7-fold symmetry applied, right? However, when you think about our drug, it's an asymmetric molecule, right? So you have an asymmetric drug molecule, which is binding to a symmetric protein with C7 symmetry. So the way I see it, this should be processed with no symmetry applied because your molecule of interest is not symmetric. So that's what I did. All the processing was done without any symmetry enforced. So this is what we called a C1 reconstruction. The first problem we had is all your compounds are fairly insoluble, and you have to have some DMSO to get enough, right? To get basically some sort of excess of your drug over the protein. However, for cryo-EM and for freezing grids, DMSO, it can cause issues. It can cause issues with freezing. So it is not recommended to exceed 0.05%. We went slightly higher than that to 0.075, and that was okay. But this is not value we can exceed a lot, so we cannot get like indefinite amount of drug there. So what we try to do is to get about 5-fold excess of the drug over monomer of Panx1. Right, so our drugs are still in excess. If it's like 1 to 7, it's even higher excess, right? Or 2 to 7. So that's as much as we can basically put in our sample due to the solubility of the compounds. So this is our test sample. It's just the DMSO, right? At 0.075. We also get compounds 35 and 62. And this is our side view. And I'm showing it to you just to show that they all look quite good. They all look like frog Panx1s. There are no significant changes. In our control, you see the map is colored by monomers. And with the addition of 35 and 62, they look almost the same. Even though we didn't enforce any symmetry, all our maps have a resolution of about 3 angstroms, which is comparable to what was published with symmetry enforced. Right, any questions? No, all right. So now let's look at those tryptophans. All right, so here is our DMSO. And as I said, there is almost always something around those tryptophans. There is always this noise. And I think I've seen it for every single map we process. Like I said, our assumption is it's just something that co-purifies from the cells. When you look at 35 and 62, the blob is still there, but it becomes bigger and more spherical. However, it's still a blob. So we are not getting anything of the quality where you can actually fit your compound in a way where you actually know 100% how it binds. There is something there, but it is I think better than what was obtained for CBX, but it's still pretty low resolution. So here is our tryptophan rings. We are looking from outside of the cell through the extracellular opening to the channel. So we are looking outside in. We see some kind of diffuse density here with the MSO, and with 62, it becomes more compact, and it seems to contact the tryptophans at least in 2 points. However, if I was reviewer, someone tried to publish it, I would have questions. Let me put it this way. So, and that's the other view from inside out. There is density here. It seems to be contacting the tryptophans, but it's very, very blobby. All right. The other thing that happened, and it happened only with 35, and I truly do not have an explanation about it. From one data set, we got two forms. About 50% particles is in what we call the active form, the inactive form. That's what I just showed you. But the other 50% have become activated. So you can actually see very nice N-terminus inside the pore. And I truly do not know why. It might be that this compound 45 has an additional effect on the protein. It still seems to, like, the density around the tryptophan 74 is the same for both. You are just getting this additional conformation with 35 that you do not get, for example, with 62. My interpretation is that every single time you see this activation of the protein, it usually happens when the hydrogen bonding network in the cytoplasmic domain is disturbed. And that is causing the N-terminus to flip inside of the pore. It might be that this compound also has like a secondary binding site somewhere here, and it's causing this effect. I don't know. It might be of no consequence to the actual inhibition, because if this is plugged, like, it doesn't matter if the rest of the protein, like if you have N-terminus inside or not, right? Like, it's plugged, nothing will go through it. So, any questions? Eva looks extremely confused. I'm confused too. Yeah, I have no explanation. Yeah. Others, yeah. They're not that different chemically, right? So, yeah, I'm also not sure why this is happening. Like I said, it might not be of a real consequence to your inhibition because both forms seem to have that plug inside. So they might both be like unable to actually, release anything because they are both basically inactivated by the compound. So it might just be an interesting observation, but I thought I'm curious about that because with probenecid, exactly the opposite was seen, right? We are going from active to inactive, and here we are going from inactive to like partially activated, which is interesting. All right.
I guess
So
one thing that I would say is, I mean,
Yeah.
I know we keep seeing this N-terminus inside, outside,
Yeah.
and all of that. And I'm not, I mean, I realize something must be making it happen, but I'm not convinced that it's really a representation of activation or inactivation.
Yeah, that's why the
I know you have the
quotation mark.
Yeah,
That's why how we say it.
we
Yeah,
put
but
air quotes on that, so we
yeah,
should just be
exactly.
mindful of that, I think.
Exactly. Yeah. So yeah, hence the air quotes. Like, that's how we are calling it for our sake. I should have probably explained it, but we don't understand whether this is like real activation, right?
Yeah, and just a reminder from Eva's work that the N-terminus doesn't seem to matter in terms of being able to activate the channel by either caspase cleavage or receptors, right? So yeah, it moves around.
Yeah, like
clearly it moves around, but
that's a very good point. Like I said, I don't know if it actually means anything. But it's interesting that we only see it for one of the compounds, not for the other. All right, so what are we going to do about it now? So I'm still working on it, so this is quite preliminary. I am hoping maybe to get the density slightly better, but I would not, from this data set, I wouldn't expect it to get a point where we are 100% sure of the conformation of the drug inside. Because what I think is happening And it's happening because the way we process cryo-EM data, right? So our protein is symmetric. It has 7 identical subunits. And during cryo-EM processing, we extract individual particle images from micrographs, and then we stack and align them together. So how are they aligned? They are aligned to the most prominent features, which are transmembrane helices, the cytoplasmic domain, and so on and so on. Between different monomers, the conformation of the drug might be quite different, right? However, this is such a small signal compared to the bulk of the protein that it basically gets averaged during processing. So that's just the consequence of how the data is treated and the fact that we are not looking at one molecule. For this one, I looked at like 200,000, 100,000, right? It's a very big data set. So the one idea that we have is to force it to be asymmetric, right? So basically, imagine, let's assume this is actually what's happening, right? So we have our compound 35. We have one molecule, right, per one heptamer, and it only can contact 4 tryptophans at a time. If we mutate remaining 3 of the tryptophans, what we are going to do is we are going to force this compound to only bind in this one conformation. So our idea was to use your concatenamers to make this type of Panx1 channel, which has a certain number of tryptophan mutations. And hopefully that would force the compound to only interact with it in 1 or 2 ways. And that would remove the problem of basically totally averaging this density as we are doing our data processing. Like I said, it's basically the question of averaging and losing the signal, because the signal from this density is very, very small compared to the bulk of the protein. That's the idea that we had. We were like curious what you think about it and what else, what else you would like us to do regarding this particular project. Yeah. So basically, I would make constructs with like increasing number of tryptophan to alanine mutation and see how many can you mutate and still get the binding, right?
Yeah, I was going to say you would want to run this through your
Cell assay, right?
your topro assay, right? Because that would be the most convenient, fastest sort of throughput way to make sure that that you still have function and that you can still block it.
It doesn't need to be frog.
Doesn't-
If
yeah.
it's human, that's perfectly fine. So I didn't talk about that. We also worked on human Panx1. It's a little bit more, like we have to use hexels to express it, so it's a little bit more expensive. It's not as stable, but we are getting like 2.7 action structures from it as well. So like we know how to work on it. We almost always start with frog just for convenience. But if we decide to actually go through this route with concatenamers, I wouldn't waste our time on frog. I would go directly to human, because that's what is most relevant here. Yeah. Yeah. So that's what we did so far. So we are currently having, like we are basically waiting for more funding to arrive in the lab at this point. So yeah. So we got a good score on 1 grant, so that should be fine. Yeah, I assume you are watching the news, right? So you know what's the situation in US. Yes. So we cannot basically promise you that will be like that's like the first thing we will do. Um, Open up the structure entirely.
Oh, that was S346D.
Yeah. So
Right. So that spreads a
so
lot.
basically, so I didn't like what you see here. That's like one of the active states we observed. But for some mutation like S346D, it does get wide, but you also completely like lose the density in the cytoplasmic domain. So it does look very strange. Um, yeah. So the with S346D, the problem is with expression. It seems to be like constitutively open, and the cells they don't like it. They die. So it's difficult for us just to get enough, right? To the cryo-EM, to do the biochemistry. So that mutation is a little bit difficult to work with. Like, and I actually don't think we need it, right? If we are going just for the drug binding, you know, wild type is fine, right? So what we actually care about is the tryptophan ring. You know from your cell studies, you work on this mutant, you know that this mutation actually, you know, the binding goes down significantly, so it must be the spot. So yeah, that's the one idea that we have.
Well, actually, can I just ask a question about that?
Absolutely, yes.
Because I guess you do your binding with the purified protein, but it is not actually in an active state. But all of the functional data are on an activated channel, right? It's either, I guess you do some kind of cleavage, right, Eva? What did you do to activate it? It's caspase cleavage. So I know you don't see any difference in the tryptophans structurally in the cleaved state. So that's the counter argument that you're making.
Okay.
I guess is there any possibility that I don't know. So does your drug access the pore or the tryptophans from the inside or the outside? And if it's from the inside, then maybe the shape, the wider shape or something gives better access. I guess I'm just wondering if there isn't some
Yeah.
reason to think about looking at it in a quote unquote channel activated by a mutation or by caspase or by
Yeah.
application at the
So
caspase site.
yeah, in our hands, we are of course we are in detergent, right? There is no cell. So in theory, anything can bind either going through the pore or going the other way, right? Like we can go, something can enter this way or something can enter this way, right? Which is different than what's happening in like cell-based assay, right? When the protein is actually in the membrane.
Yeah, but I guess the drug, the compound,
Yeah.
whether it can actually get there
Mm-hmm.
is going to be determined by the shape of the protein. And
Yes.
the fact that the protein might have a different shape when it's activated versus inactivated it could still affect drug access even in your purified state. I guess is what I was sort of.
Yeah. That is true. If it's in the active state, we also see these like side openings here between subunits, which they get quite big. On the other hand, like the size of the tryptophan ring it's constant, right? Like it's a possibility. But I think like when we are dealing with purified protein, which is in detergent, access is not that big of an issue. What we do is we incubate the protein with the compound, at least for cryo-EM, overnight. So we are really hoping for it to get saturated, if that makes sense. I don't know if I'm answering your question, Doug, but I think this is more
Yeah, no, I think all of
of
that
a
is.
concern in cell studies.
No, I mean, I understand what you're saying.
Yeah.
I understand. Yeah, you saturate it, you leave it overnight, all of that. But I mean, if the thing can't get there, it can't get there, right? And
Sure.
so, and that can be a function of the shape of the protein, no matter how long you leave it or how high the concentration is. So, all I'm saying is the functional data are all done with a functionally activated channel, whatever that looks
Yeah.
like.
Mm-hmm.
And to the extent that it might interfere with access, it could change what you're able to see in this assay, in this structural assay.
Yeah. On the other hand, with 35, we are getting this activated state.
You're getting
Yeah,
what?
it's activated. Well, at least the N-terminus is inside and but it's
Yeah.
not splaying the channel as much as K140R
It
or
is.
S346D. The splaying is not as big.
It's the
is what
same. It's the same. It's the same.
But what's interesting is you could activate it with caspase and you see no splaying of the channel.
Yeah.
Yeah,
So like,
this is the huge confusion in all of
Yes.
this, right? So I mean,
Yeah.
I hear you, and I think, I mean, I'm not advocating anything in particular. I'm just sort of, making a caveat here. That there is,
okay. So I'm thinking how to get it for cryo-EM. So even before we, do the assay to be like fully activated. Like how to purify it, how to do it, right?
Oxidation.
It's something more physiological, I would say.
I know, but it's the easiest
sorry,
way.
so I'm just complicating things here. Maybe the simplest thing to do is first to
No, but
try.
it's a very good point. Like it's possible that we will actually see something different, and I think it's actually good to at least think about it, because concatenators are like insane amount of work. So even if we actually go this way, I think before we start, it's good to have this discussion, right? Maybe we can do something smarter than that, or at least try.
You could try with the S346D.
Yeah, but what Doug is saying,
Is access to the ring.
so with S346D, what's happening is the protein gets quite wider in the cytoplasmic domain, and we are also losing a lot of this density for the C terminus, right? So one of our ideas was if C terminus is regulating the access, well, basically when it becomes disordered, like the access is much easier. So that's one thing.
We have to remember that there's a whole C terminus below that we don't see, right? That's totally disordered. We never see it in our maps.
Yeah.
So we don't know what it's doing down there, actually.
We know that we can absolutely do S346D delta C, and that's like as open.
That's what I was just thinking. S346 delta C is an activated channel, passes lots of current, makes dye uptake like crazy, and you have it already.
Yeah.
And you already have like really nice maps of it.
Yeah, we got it like 3 angstroms, and yeah, that's one possibility, because like here it's as accessible as you can probably make this protein accessible. Right? You completely remove the C term, you open it up, and we know from the physiology assays that everything goes through. So, yeah. Maybe. But even with that solubility, we were able to get it in like 5-fold excess over monomer, which it might not be fully saturated at this affinity, but I think you should be able to like see it if it's there, right? But yeah, of course, having it more soluble, it's always easier. But I don't think that's like the main problem we are facing here, right? I think it's there. We sort of see an indirect effect of 35, even though we don't see the density, right? Because we see a change in conformation. So I think it is bound to at least some of the protein particles. Like I said, I think this is just a consequence of how the data is averaged and processed for cryo-EM. That it's basically right on the symmetry axis, it's binding slightly differently to all the different proteins that we are using in this analysis, and the signal just gets lost. And we see it all the time. We know we have our protein in micelle, but we don't really see the micelle very well. We see just a very weak shape. Same goes for the C term, right? We don't see big part of it. We can see like a low density plaque, but the rest of it, we are not able to resolve. So yeah, I think we should have a think about all the different possibilities. Like cryo-EM analysis on this, it's going to be tough. With connexins, like some of the slides that I showed you before, like that group was actually able to get EM structures of these compounds, but they bind very differently. It's like 1-to-1 binding. You get connexin is a hexamer. You get 6 different compounds bound per channel, and they sort of bind like inside the funnel, not at the opening. So that was much easier. I think Alex.
He said something in the chat about
Increasing solubility.
Yeah.
Mm-hmm. Yeah.
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 Well, actually, at the beginning, I think we used the leasing. But however, if you think I don't remember which compound, like the 62, or you just tell me the one compound is show like the 0.24 micromolar. If that one is good, and can prepare for you, because we need to try to use like the ammonium counterion, just try to make it more soluble. We can check the solubility in our site. So that's maybe help you to get the better resolution.
Yeah. So yeah, that's what we have. I know it's quite late for you. Yeah.
Yes, no, because I have the class early class at the Tsinghua tomorrow morning.
No, yeah. Thank you so much for
That's
being
okay. I
able to
still
meet. I know it's 12 hours
can
different, so it's
listen.
tough. Yeah. I'm anxious. Yeah.
And excuse me, I want to ask a question about
Of
the
course.
KD part in compound 62 and
Mm-hmm.
compound 72, compound 70, because the KD is very different.
Different.
The KD
Yeah.
in compound 62 is very low, and
Yes.
however, in my diptych assay, the compound 70 and compound 62, the inhibition is almost the same, I think. So
Yeah.
I
So
want to ask you, do you think which one is better for us to use in research or in
yeah,
our?
don't use this. Don't use this. Like this is very preliminary. first thing, it's not a human protein, right? It's a frog protein. So that might be the first source of the difference. Two, if you actually look at the curves, especially like in the early points, they could be much better, and we will try to make them better to actually get those binding curves better. So before we actually get that done, I wouldn't use this data to guide your assay, if that makes sense. And also, like the fact that some values are going to be different, I think that's the nature of two very different experimental setups, right? You are working on living cells, and this is completely purified recombinant protein, which is in detergent. It's no longer in the cell membrane. And we are just adding more and more drug to it. So I think right now we are sort of seeing the similar trend from both of those assays. Not identical, but similar, but like treated as a preliminary result, basically, if that makes sense.
So I have a little question
Of
about
course.
I just wondering that, so now these data use, we know that compound 62 have a poor solubility. So
Yes.
what's the situation when it do the experiment? It's solute
Yes.
completely or not?
Yeah. So we were able. So this, I didn't mention that, but this assay was done in 5% DMSO.
Yeah. Okay.
So frog Panx1, we did the stability test, and it seems to be okay in even quite high concentration of DMSO. I
Yeah.
think we checked up to 15%, and it's fine.
Yeah.
So at this concentration of DMSO, if we go to 200 micromolar, we do not observe precipitation. If we use fresh compounds,
Yeah.
what we observed, and we actually don't really understand, like if you make your DMSO stock and you
Yeah.
leave it even for a few hours, it starts to like precipitate out of the stock.
Yes, I know.
Yeah, like we tried to like sonicate it, heat it up, do all of those things,
You,
and it's like it doesn't care.
yeah.
Yeah, but if you have DMSO in your buffer and you start with freshly made stocks, it seems to be fine to this concentration.
Yeah.
However, what I'm also not sure about, and I think Doug pointed it out before, this saturates at much lower values than the other curves. And I am not sure why. It might interact with tryptophans like slightly differently, or it might actually have to do something with the solubility.
Yeah.
So that's also something to consider.
Yes. Those. Okay, I think the solubility might be
Yeah.
issue
Okay.
that affect these data's
Mhm.
true or yeah. Okay, and we will change the ion channel of our compound to
Mhm.
make the compound more soluble. Yeah. That's it. Thank you.
Absolutely. All right. So from our side, that's all we have accomplished so far. Yeah, I don't know if you guys want to think about it, meet at some later point of time. As before 6D. Yeah. 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 Let's maybe, because like I know from Susan's work how difficult it is. So what would be like before we actually commit to concateners, it it would be good to have data where we have a little bit more information how the drug actually interacts. Yeah. Yeah, because like even yeah, yeah, exactly. So that's the idea that I had, but like you said, I don't know how much cleaner the cryo-EM dataset is going to be. Because even if you remove those free tryptophans, you can still imagine that that will bind into different conformations. It can bind like this, and you can also flip it like 180, right? And it will still bind. Maybe. Um, so that's something I will keep thinking about it, how to approach it. But yeah, I understand this is some work, right? It will take some time to actually make those, especially that you will have to like test like the progressive tryptophan deletions. Yeah. Um, so yeah, uh, we can discuss here in the lab about possibly trying S346D. If you guys can have a slightly higher soluble, more soluble version of the 62, that will also be helpful. And based on the results, we use concatenators or not.
Yeah, we're building concatenators, as you know, Eva. I've been
Yeah.
pestering you about questions. So now we've kind of altered them. We've eliminated the C-terminal caspase site because we find that when we grow it in the hex cells, we get endogenous cleavage. Yeah, and the amount of the inhibitor that we would had to add was prohibitively expensive compared to what you guys had to add for a single cell. And then we also wanted, when we're trying something where we want to be able to tag the first monomer
Yes, like
of
know
the
which one is
concatenator,
first.
so we know where we are in the map. And so, well, the last one, well, because we got to build everything from the beginning, it seems to be easier to do the first. And then, because this is also for the K140R and the S46D, when we build it, we want to, the K140R is very subtle in terms of its effect. So we need to know, okay, where did we stick it in the concatenator? And so it would be easier just to start with where we know monomer number 1 is. And then I can say, oh, monomer number 2 is wild type, or monomer number 2 is K140R, or whatever. So we're sticking a strep tag into an intracellular loop based on our maps. And then we'll go back at it with a modified streptavidin, and we hope, purified streptavidin, and we hope that it will tag our first monomer. And then we can say, then we can count after that. And then, like Eva said, we have to do all of this in C1. So we need some really good, clean, purified protein.
Oh.
Oh, Doug can't hear anybody. Closed captioning is working. Oh, okay. I didn't even know.
It's CC. It's CC. Yeah, I found it. Just click turn on captions.
Oh, yeah, I saw that commercial. Yeah.
Mm-hmm.
So I've just inserted the modified strep tag into the PC, right, the pcDNA plus 1 monomer. So I inserted it into the first monomer. I got to sequence it, make sure that's okay. But then I'm ready to start building. Then I'm ready to go. That's what we were. Yeah, well, we kept changing our mind, like, okay, once we determined that the caspase inhibitor wasn't going to be feasible, then I went back and modified in PEBB, the building block. I modified that, so I changed it to either factor X, that site, or we just changed the DVVD to AVVA. So that was done. And then I was like, okay, I'm ready to start. And then Eva was like, we really need to tag. I mean, Ega was like, we really need to identify where we're starting. And so then we went back and determined that we think we can go into intercellular loop 2. There's this junction that we've identified in our maps and put a strep tag. And then I think today just arrived the modified streptavidin that we can then purify and then add to our. So I think it's just about ready to start the building process. Well, I think we talked about that before, and I said, well, I guess I can do the last step that you described in the Nature Com paper, if we want it or not. Like right now, I was under the assumption that we didn't want the EGFP at the end, but we can do that. It's just like the last step that you did. Yeah, so that's why you like it and not a coach. Yeah, you don't like the co-transfection strategy with another. Okay, yeah. So we can talk about that and add it at the end, like you did in the or Sammy did. You made the 7 MERS, right? You made the 7-7, and I think you did, and that was. I looked back at the dates, and I was like, wow, it was like 2015. Yeah. Yeah. And they're coming back big time.
Can you hear me?
Oh, there you go.
Yes.
Okay, I can't hear you, but I don't have any more comments. I've been reading along.
Mhm.
Down for it.
Yeah.
Yeah, we just talked about it this morning, and we actually talked about the S346D delta C because we do have virus of that, and the cells are okay. So I think that was we had 3 that we were going to be growing. Why? No. So that's just what I don't get is in the full length, it's a pain, but in the truncation, it grows a lot easier. So yeah, I think I remembered. Yeah. You did those. Yeah. 57. Yeah. So yeah, if the virus is here, why not grow it? So we'll grow some and we'll see what's going on with that. But yeah, I was surprised that grows better than the full length. I don't understand.
I have a last question
Yes.
about the. I want to. May I ask that how much amount the compound left
We
there?
have a lot. You gave us like kilograms. We will
Not that
never
much,
use the whole
bec-
thing.
because I just wondering that I just need to take time to synthesize the other
Yeah.
counter iron to compound. So
Yes. We don't
maybe
need
the
that much.
maybe the compound in your hands that it's and I will send you the new one for with the new counter ion.
Sorry.
Okay.
1 second.
Okay.
Oh, by the way, hey, wait. I want to.
Mhm.
Well, you can continue.
Yeah, go
It's
ahead.
okay.
She's right
It's
there.
okay. Even you want to talk what you can say it.
Because the previous slide shows that maybe it's previous or the previous lot of slide shows
This one?
that 就是 let me use Chinese sorry 就是那个
OK
我想问就是那个前面是不是有提到就是要把 Panx1 的 tryptophan 74然后 mutant 3个对不对是这个对不对然后我记得我们之前有做那个叫什么mutant 变成 W74A 然后把那边改掉嘛可是这样子的话那个 expression 就很烂然后还有那个 adaptate 也有点做不太出来然后只是在想说，如果他们真的要做4个的话，会不会也做不出来？因为我们觉得，我们自己做完，我自己做完之后，觉得那边是整个活性或者整个结构稳定最重要的地方。
Okay. Yeah. Yeah. I'm not surprised. Like, yeah.
Yeah. Well, yeah. Hopefully we only have like 4 or 3 as opposed to 7,
Yeah.
will help. Right.
Yeah.
Right. We're making the W74A, at least in frog right now, as a heptamer, right? So it's a single monomer and then expressing that we're making the virus right now. So we'll know, well, that
Yeah.
takes 2 weeks for that to grow. So we won't know for
Oh,
that. period of
tryptophan
time.
74A mutant in human Panx1 the expression is okay, I think.
Oh,
So,
okay.
yeah. So sorry about that. So basically, this is. So this is 74. The tryptophan ring around the extracellular domain is 74 in frog, but in humans, I think it's also 5.
It's off by 2. It doesn't change until later. So it should be 74.
So okay. So this is what we are basically interested in, like the side chain, but it's lining the extracellular opening, right? But like
Side chain?
yeah. So this is
Well,
another
actually, there's a
interesting
paper
thing.
that argues
Yeah.
that out there.
Yeah, and
Yeah.
that tag like the positive charge or negative charge diaptech is difference in tryptophan 70 or A mutant PNC1.
Yes. So at this resolution, you should be able to see those tryptophan chains very, very well, but they don't appear to be that well resolved. And we've seen it consistently in all of our maps. And some groups argue there is flexibility to the side chain. There might be different rotameric states, and that's what might be helping with actually transport of the different compounds, right? Because if you change their flexibility, maybe larger things can pass through easily. Interestingly, with the compounds, they actually do look better. It's like they are slightly stabilized, but it's a subtle difference. So yeah. So, arguing in a paper when you're trying to publish something about all of those subtle differences can be challenging.
Could you do focused refinement, Ego, in that region or no?
I tried, it doesn't help.
Doesn't work? Okay.
Yeah. So yeah. Something to think about. Yeah. But like you can think that if you remove this constriction, things will leak from inside the cell out, and the cells might be quite unhappy about that and growing more poorly.
More holes in the membrane.
Yeah, basically, yeah. All right.
All right, so it's got to be late for you guys.
Yeah,
It's almost midnight.
absolutely.
Yeah.
Yeah. Yeah, absolutely. Yeah.
Yep.
It was very nice to meet you, and we'll be in contact. Okay. Have a good night.
It was nice meeting you
Bye.
guys, the rest of you.
Thank you.
Eva, I know you.
Reading along.
Bye. Yeah.
Bye.
Bye.
老师那个可以翻译，没有，我只是要跟学姐确定一下我有没有听错而已。我在问那个，就第一个就是那个 KD 不一样嘛，然后我一直在想，到底真的哦，然后所以对对对，我知道是两回事，但是我刚刚一直在纠结说它的 KD 差这么多，然后到底，OK OK，然后还有就是，所以我要试着去做那个，对，我刚刚也在，我也在对吼，I got it，yes。然后再来就是那个，所以我要做那个叫什么 point mutation 那个，我真的不知道要怎么让7个里面只变4个，哎，他们做，那就好，哦，OK OK，那所以暂时还没有，嗯嗯嗯，OK，好，所以暂时还没有。OK，那所以还没有，暂时没有我的地方要做。我以为那 mutation 要， 嗯嗯，是的，哦，就是 Panasonic 某个 side 从 C 变 S 之后，它会变 active f o c 这个意思吗？哦哦，这么酷。OK OK OK，嗯嗯嗯，OK，对，嗯，OK OK，嗯呀，OK，嗯嗯嗯，说可以用 primer 自己在那边，嗯嗯嗯嗯嗯嗯，OK。嗯，好，那所以我后面 Sensa 一样就是用 compound 70，因为我刚刚问那个问题是我很怕就突然放个60个在做。OK OK，对，因为我刚刚听起来是这个意思。哦，OK，对，刚刚问的呀，嗯，OK OK，嗯嗯嗯，什么5% DMSO，嗯，哦，嗯嗯嗯嗯嗯，OK，上去，嗯，OK OK，我要在意这些。OK OK OK，好，那就好，那我就继续走下去。然后我还在想什么时候要做那个什么 Frog Panasonic Full Lens 那个 GFP 的那个，我要把它 mutate 回来那个。等我下次要做那个的时候，我再顺便设计 maybe 我们刚刚讲到那个 mutation 的那个 Panasonic 的那个 primer，或我下次要做 mutation 的时候，我再顺便 P 过去。反正如果不 work 就算了。嗯。OK OK，我稍微把这件事情记一下，下次做 PCR 的时候再顺便用它。那我就继续，当然会，所以它是一个疾病的一个 form 的 penicillin 吗？因为我知道有什么你们的人，嗯，OK OK，嗯嗯嗯，哦 好，OK，到时候我要做的时候再跟老师确定一下，因为如果是他是那个疾病的那个，我应该知道是什么 R140还是什么 H 下面也，我记得，嗯，很有趣的。OK OK，我只记得那篇，我有看过那一篇 paper。哦，嗯嗯，好，OK OK，那我就继续加油做下去。哦哦，那就好，抱歉，那就好。OK OK，好，嗯，对，哦，哎，他不是，我就，哦，对，我只大概抓到他，好像叫我不要理他，然后是 frog 跟 human 就这样而已。哦，好，OK，好，好，拜拜。那录影，哦哦哦，不会传出去吧？啊，哎呀，啊，没关系啊，他们听不懂，啊，没关系，好，拜拜。